Welcome to ICANX Talk, Connecting the World and the Universe. I'm your moderator today, Professor Martin Thuo, coming to you from North Carolina State, Eastern United States. This month, we continue our theme on bioengineering. We heard from Peter Wang, Johan Foster, Lindsay Warren, and today we are privileged to have the son of Africa, a great scientist from the continent, Professor Kelly Chibale. Today, I'll be assisted by three panelists, our ICANX challenger, John Woodland, Ndikoma Olugwem, and Adrian Atkins. Now, a little bit about our speaker. Professor Kelly Chibale is originally from Zambia. He has a fascinating story, a story of triumph, a story of determination, a story of dedication. He grew up in rural Zambia, where, according to one of his interviews, he used to go to school without shoes and rose to be a global scientist. From there, he went to UK, where he worked to Cambridge specifically, and worked with the very famous Stuart Wallen, where he was working on natural products and medicinal chemistry. From there, he jumped the Atlantic and went to work with Casey Nicolau at Scripps Institute. And there he was involved in working on brevetoxin A analogs. From that point, he returned back to Africa and ended up in South Africa, University of Cape Town, where he started building his own group. First, he tried to do photosynthesis of molecules, and the resources were not there, so he switched gears and reinvented himself as a medchem, and he has been very, very successful. He's the chair of South Africa's research discovery. He has many awards. I'm not going to go through all of them, but the most prominent one is being the director of the H3D labs at the University of Cape Town, from where he'll be sharing some of his work today. He started out as a lecturer at the University of Cape Town and rose through the ranks to being a full professor. And he leads a decent-sized group out of that university. He's in the board of several journals, one of which is Angvante Chemie, where we serve together. He has also very well published, and just to give you an idea, in 2022, he has published 15 papers, 2 of them in premier journals, Nature Medicine and Nature Chemistry. I don't want to take more of your time, Kelly. I want to hear about drug discovery, so I'm going to hand it to you. Thank you. You can share your slides.
Thank you, Martin, for that very kind introduction. And I hope that you can see the slides here now.
Yep.
Thank you so much, everyone, for coming to listen to my presentation. And I'm very grateful to ICANX, Martin and Alice and colleagues for giving me this opportunity to share with you what we have been doing at the southern tip of Africa here in Cape Town. This is the outline for my presentation. Firstly, I will introduce our Drug Discovery Center, and then I will switch to talk about translational medicine so that we have a perspective, a context of how this terminology is used in the context of the project I'm going to be describing. And then thirdly, I will describe a journey that we took on one aspect of translational medicine, and also discuss with you how we deconvoluted the mode of action to understand how the compounds that we discovered works. And then finally, from the insight gained in the clinic, how that informed our strategy to develop backup candidates. And then I'll finish off with some takeaways. All right, introduction to H3D. H3D, the H in H3D is holistic, and of course the 3 Ds. The first D is a drug, the second D is discovery, the third D is development. We were established in about 2010 at the University of Cape Town. So we are actually embedded within the University of Cape Town environment. And as you'll see in subsequent slides, this is an integrated drug discovery center, integrating the multiple disciplines that you need to execute or prosecute a drug discovery project from the lab to the patient. And it's the first and only one of its kind on the continent, and a very special setting in a university environment. The vision really is to be a leading organization for drug discovery on the African continent. For the last 11 years or so, we have been on a mission, and this mission is threefold. We've been involved in a mission to discover innovative medicines for diseases that predominantly affect our continent. And I'll give you an example of that in the context of this presentation. We've also been on a mission to develop preclinical tools that will allow us to prioritize small molecules during their discovery and optimization based upon their predicted pharmacological profile in African patients. And thirdly, but not least, we are really involved in education, training the next generation of African scientists who are really skilled in integrated drug discovery. We have made enormous progress over the last 11 years or so, and this year marked a very significant milestone when our center became one of the 3 Johnson & Johnson global satellite centers for global health discovery. This is a satellite center focused on global health discovery with a specific focus on addressing antimicrobial resistance, more specifically, precision antibiotics, aiming to develop first-in-class multi-drug resistant gram-negative therapeutics. This is very significant, not only for the African continent, but globally as well, to really give a perspective that this is only one of 3 centers globally that the company has set up. The first one launched in London last year. We got launched this year in April, and in June this year, another center was launched in Singapore. So we're not just focused on AMR or antimicrobial resistant or antibiotic discovery. Disease portfolio, infectious disease portfolio also includes malaria, tuberculosis or TB, AMR as already mentioned, and the program on coronavirus. And this is all supported and underpinned by technology platforms, which I will share on the next slide or so. And of course, these, on top of the scientific programs, are our current funders, ranging from the pharmaceutical industry to our government. Really significant support from the government of the Republic of South Africa. So in terms of the drug discovery platforms, which I referred to earlier as an integrated platform, so we have the chemistry platform, the medicinal chemistry platform. Along with that is a computer-aided drug discovery or CAD platform, including an artificial intelligence machine learning component to really develop models using experimental data that we've accumulated over the last 11 years or so. Chemistry is one platform. The second platform is a pharmacology platform, including our drug metabolism and pharmacokinetics platform, DMPK. We have a suite of in vitro absorption, distribution, metabolism, and excretion, or ADME, assays, ranging from solubility to microsomal metabolic stability across different species, rodents and humans. And we also do our own in vivo rodent pharmacokinetics in-house. And that is the final component of this, is a biology platform, again, reflecting the disease biology and the therapeutic areas of focus in the center. One is the malaria biology, both in vitro, but also we have a humanized mouse model of malaria infection that we use in conducting efficacy studies. We also have TB biology, and we do antimicrobial resistant screening on several pathogens that have been listed as important by the World Health Organization. We also have an enzymology platform to support target-based drug discovery programs, and also cytotoxicity cross-screening to ensure that what we're developing is likely to be safe. And of course, when you have such a huge program across multiple programs and therapeutic areas, data management and data storage becomes very critical. All right, the message on this slide is that everything we do is really about partnerships. And I don't want to go through the intricate details of the partners here, but it range from the pharmaceutical companies to product development partnerships, like the Medicines for Malaria venture, to academic institutions. And of course, one of the other important things about product development partnerships is that they really provide a mechanism to bring the private sector and the public sector and academic institutions together. Two examples of these product development partnerships. One to the left of the screen here is the TB drug accelerator, and to the right is the malaria drug accelerator. But these are really international consortia supported by the Bill and Melinda Gates Foundation to facilitate early stage drug discovery. This is all pre-competitive drug discovery, that we share data, knowledge, and expertise across members of the consortium in a pre-competitive space. And of course, this is a very powerful way to move programs forward, to share resources, share expertise. And that's really is critical to what everything else that we do. Let me switch now to just providing a context of the word translational medicine, which can mean many things to different people. But the best way to understand translational medicine is to understand traditional medicine. And I've put this in inverted commas, traditional medicine. It's not what you probably are thinking of, the stuff we use in many parts of the world. But this is really the definition of traditional medicine in the context of translational medicine that will come up in a second. So what you see on this slide is that the bench to bedside, the fundamental difference between the bench side and the bedside is that on the bench side, the focus is on developing a pill, a medicine. The focus of the bedside in this traditional medicine context is on treatment. Keep this in mind, because translational medicine doesn't have any of these focuses here. Translational medicine is really about moving from the bench to the bedside, underpinned by scientific insight, and equally moving from the bedside, from the clinic, underpinned by clinical insight, back into the lab. So everything I'm going to describe from now onwards, this is the context of translational medicine. I will share with you a journey of moving from the bedside to the bedside, underpinned by scientific insight, and also moving back from the bedside, from the clinic, back into the lab, underpinned by what we saw in the clinic. And this is the program that I'm going to describe. But before I do that, let me give you a very brief introduction to the unmet medical need of malaria. Drug discovery is about addressing unmet medical needs. Malaria is a significant issue, particularly on our continent. You can see this map of the world. What's really shown in red is, of course, the geographical regions of the world where this disease is endemic. It is a disease caused by various species of Plasmodium parasites. And the most significant of these is Plasmodium falciparum and Plasmodium vivax. And of course, my continent of Africa is really arguably the most affected of everybody, responsible for a huge number of deaths. And of course, significant negative implications on the economies of this continent. And of course, unfortunately, the vast majority of people that suffer from this disease are children. Arguably, the most significant issue to either existing medicines or new medicines or future medicines is drug resistance that has rendered, not just in malaria, but in many other therapeutic areas, existing medicines useless and diseases impossible to treat where that resistance is prominent. So this is just so that you can keep in context that we're dealing with a disease caused by a parasite. And of course, the resistance is in the context of parasite resistance. What are we looking for in new medicines for malaria? The number one priority for this disease is circumventing drug resistance. And to achieve that, we've got to be able to identify novel molecules that function by novel mechanisms, something the parasite has never seen before. That's the most urgent priority. But the other additional benefits that these days, it's no longer good enough to come up with a malaria medicine that only relieves clinical symptoms. We're looking at new molecules for malaria that work through novel mechanisms that are also able to kill the parasite at multiple stages. So if I walk you through this diagram here, so you see a little mosquito. This is the female Anopheles mosquito. During a blood meal, when it's sucking blood from a human host, releases these parasites we call sporozoites, which get into the bloodstream. And the first port of call for the sporozoites is the liver. When the parasites are in the liver, the symptoms of the disease are not evident. And then, of course, the multiplication begins before the parasites infect the host red blood cells. The malaria caused by Plasmodium vivax, some of those parasites go into dormancy. We call them hypnozoites. So these are dormant forms of the parasites that only become an issue where you have relapse due to a number of risk factors. But otherwise, the journey moves to when the parasite infects the host red blood cells. And this is the stage where the clinical symptoms of the disease become evident. And during the so-called asexual reproduction of the parasite in the host red blood cells, there is another process that goes on, which is one of a sexual nature, sexual reproduction, which involves what we call the transmissible forms of the parasites. We call them gametocytes. And so what we're looking for in new medicines for malaria, in addition to circumventing drug resistance, we're looking for molecules that can kill the parasites when they are in the liver. And something that's missing here is the word chemoprotection. We can protect patients if we can kill parasites when they are in the liver, when the symptoms are not evident. So we can offer protection. If we can kill these dormant forms of the parasites caused by the disease caused by Plasmodium vivax, we can combat relapse. And this is the protection. This is actually in the wrong place. This protection should have been here where the liver is. Finally, if we kill the parasites when they are in the blood, of course, we can relieve clinical symptoms. And also, when we kill the transmissible forms of the parasites, the so-called gametocytes, we can interrupt transmission of malaria from person to person via the mosquito. So this is the picture that you should keep in mind as I walk you through the rest of the presentation. This is the story that actually started in 2009. And here is a good opportunity to just remind you that there are two complementary approaches to drug discovery. One approach relies on phenotypic wholesale screening to screen libraries of molecules for phenotypic wholesale activity, which is then followed by mode of action deconvolution to understand how the drugs work. The second approach, of course, is one where the target is already known, and we can then screen libraries to derive inhibitors of that particular target that we then optimize with respect to safety and efficacy. This approach, this project, the story I'm about to share, is a phenotypic wholesale screening approach. And the starting point was screening of a commercial library. Then the company used to be called BioFocus, which was recently acquired by Charles River. And this particular library we screened is known as a soft focus library. Soft focus libraries are designed with a particular gene family in mind. And this could be, in this particular library of 36,000 small molecules, we had sub-libraries of different gene families. For example, kinase gene families, G protein coupled receptors, ion channels, etc., etc. This library was screened phenotypically on this drug-sensitive strain of the human malaria parasite, Plasmodium falciparum, and a drug-resistant strain, because the challenge is arguably overcoming drug resistance. This same library was screened in parallel on mammalian cells, and we focused on selecting sub-libraries which showed inhibition, and this was a cutoff of at least 80% inhibition of parasite growth at this concentration, but also selective killing in terms of selectivity over mammalian cell lines, which then gave us, using this criteria, more than 200 potential starting points representing different chemotypes. The ones showed here are just examples of the top 4 that we prioritize. What I'm going to share with you today is just an example of how we prosecuted one of this series from this program here as an example. So this program, when you start a drug discovery program, like in this case, before we could even start, we had to understand what is the target product profile, which then informs the target candidate profile, so that when we develop assays or a screening cascade, it is informed by the end goal. And the end goal is about the patient. And in this case, when we think about malaria, 1 or 2 significant target product profiles of a malaria medicine, that it must be oral, must be given orally, because this is a disease that largely affects people in poor communities of our continent and elsewhere. Secondly, the cost of goods. It must be cheap. So that target product profile, the 2 examples I've given you are just examples, informs the screening cascade and the progression criteria. So the project goals that we defined from the very beginning, informed by the target product profile, was firstly, we wanted to develop an oral drug to treat malaria caused by Plasmodium falciparum and Plasmodium vivax. Secondly, because resistance is the #1 priority, we had to make sure that we front load experiments that will help us to focus on those compounds that are likely not cross-resistant with what is available on the clinic. Finally, but not least, is that we're looking for also chemical matter able to kill the parasite at multiple stages, so that we can really contribute not just to prevention of malaria or control of malaria, but also contribute to the elimination agenda by interrupting transmission. All right, the first stage of the process. So this was 36,000 small molecules screened. Using robotic systems, how do we ensure that before we invest resources in this program, that what we saw from the robotic high throughput screening was not a false positive? So the first step was to undertake what we call hit validation. So we resynthesize the molecules, we characterize the molecules, and we retest to confirm the potency that we saw from the high throughput screening. And until then, we were not willing to invest resources before we do any hit validation. When that was confirmed, we then initiated the next stage of the process. So we identify the hit, which we validated. The next phase of the process is the so-called hit to lead, which is really critical in terms of demonstrating a pharmacology proof of concept early on, but also identify issues that might be an obstacle to that small molecule being a safe and efficacious drug. So this is what we did. We do the typical medicinal chemistry, which is an iterative process to identify issues which then form the basis of the lead optimization campaign. And the first molecule that you see on the left-hand side, which is this molecule that came from the screening library. Of course, one of the things that we identified, if I go back to one slide, which is highlighted in red, drug discovery is about identifying issues so that you can then address them in the lead optimization process. So early on, there were two issues with this starting point. The first one was that solubility was not where we wanted it to be, ideally. The second issue to do with the microsomal metabolic stability, which of course was an issue that could impact on the dose and of course the clearance of this compound if it eventually made it into people. So through this iterative process to address the issue of low solubility and low metabolic stability, we identified this compound here as an early lead in the process of doing this program here. So we could see that we could improve solubility and we could improve metabolic stability as shown by this hepatic extraction ratio, EH. So the lower the number, the more stable the compound is in the presence of liver enzymes or macrosomal preparations. The one limitation with this compound in the middle here, we're actually threefold. Number one, the predicted human dose, which we front load in our drug discovery programs, was very high because an important target product profile for malaria medicine is that it must be cheap. And so when we did the human dose predictions, we found that the dose predicted for this compound here was higher and not really consistent with our target product profile. The second issue with this compound is the potential cardiotoxicity risks as really signaled by this human ether agogo related gene or HERG, which is a potassium channel. And so we had to do additional medicinal chemistry iterations to address these liabilities. And that effort, in addition to improving on the potency, led to this compound here on the right-hand side, which had not only improved potency on the parasite, but also, although we compromised somewhat on solubility, which came back to haunt us a little bit later in development, but we significantly improved the microsomal metabolic stability and clearance in the presence of liver enzymes, but also de-risked this cardiotoxicity flag as signaled by the HEG potassium channel. So just to show you a bit of data that brought excitement to the program, when we conducted the efficacy study in the humanized mouse model, this really exquisitely was very potent, delivering an ED90 of less than a milligram per kilogram. Again, really consistent with potential low dose treatment, which has implications on the cost of goods. And of course, we also found that this compound had very good pharmacokinetics, very much consistent with the significantly improved microsomal metabolic stability that I showed you earlier. Very long half-life that would really provide coverage over 8 days of infection, and therefore provide protection to the patient when they are on treatment. And of course, the clearance was very low, as we saw in rats and other species that I'll touch on. All right, so what is the profile of this? I'm gonna go back and remind you, in addition to circumventing drug resistance, it's also coming up with compounds with multi-stage activity that can offer additional benefits. And that's what we see with this compound here. I don't want to bore you with the details of this. This was published back in Science Translational Medicine in 2017, but really making the point that we had supporting data that this compound, given the long half-life and given the activity on the multiple stages of the parasite life cycle, really has the potential to offer protection, relieve clinical symptoms, and also block transmission of malaria from person to person by interrupting transmission back into the mosquito. So everything ticked the right boxes. Now, summarize this in the following way. One of the things that kills projects in the preclinical stage is safety. That's where most projects die. That's the valley of death. Many projects die because of safety considerations. So we did extensive toxicity studies, genotoxicity, exploratory 7-day rat toxicology. We did pharmacokinetics across different species, from rodents to non-human primates and dogs. And this data was really critical in doing human dose predictions. We determined the safety margins and did the predictions of what the dose is likely to be in people. And this led to a major breakthrough in 2012, when this compound was selected and approved as a preclinical development candidate. What then ensued from 2012 is additional extensive good laboratory practice, GLP, rat toxicology studies, to really give confidence about the safety of this compound before entering first time in humans. And that was achieved in 2014, when this compound was tested first time in human, which in fact was another significant milestone in the fact that, in addition to the drug discovery program that I've described being led out of Africa by an international team led out of Africa, the first phase 1 trial of a malaria candidate was also performed right here in Africa at our institution in 2014. This molecule went further into phase 2 human studies, which is not really the subject of my presentation today. All right, all along, we didn't know how this compound works. We didn't even care to know how it works. The initial focus was delivering a compound that was both safe and efficacious and met all of the initial target product profile, including safety margins, including the cost of goods, including all the multi-stage activity profiles. But we had to also deconvolute the mode of action, because if we don't understand how the drug works, when, not if, when resistance emerges, we're not going to be able to address that resistance if we don't understand how the drug works. So we took two complementary approaches to mode of action deconvolution. The first approach was a genomic approach where we selected resistant mutants. So we put the parasites under drug pressure. When they develop resistance, we conduct whole genome sequencing. And when we do whole genome sequencing, we look to see where there are mutations. And what we saw was a single nucleotide polymorphism in the gene encoding for a kinase, specifically a lipid kinase, phosphorinositide 4 kinase, or PI4K. The challenge of this approach, this genomic approach, is that when you deconvolute the mode of action here, you may not necessarily be deconvoluting the mode of action. You may actually be deconvoluting the mode of action, the mode of resistance. So what we needed to do is to really have confidence that this is not just a mechanism of resistance, but that it's also the mechanism of action. So we took a second approach. This is a proteomics approach, more specifically, a chemoproteomics approach using the so-called kinobeads technology. Kinobeads are basically used a lot in the oncology field, in the cancer field, where a number of hundreds of human kinases are immobilized on beads. And that allows one to study the interaction between a small molecule and hundreds of human kinases. This kinobeads technology has been adopted to the human malaria parasite. And about 90 or so malaria parasite kinases have been immobilized on this kinobeads platform. Anyway, using this approach, pull down and competitive binding experiments exquisitely nailed down Plasmodium phosphorinositide 4 kinase as a target, confirming what we saw from the genomic approach of doing resistance selection and whole genome sequencing. Highly selective over several human kinases. And the only human off target we saw at that particular concentration was this P1, P4, K2C, which is actually an off target of the anti-cancer drug Gleevec. Otherwise, this compound is very highly selective, and we understand the basis for that selectivity. The question is, could we correlate the enzyme inhibition that we saw across multiple series to the host cell antiparasitic activity? And we saw this really beautiful correlation between enzyme inhibition and host cell antiparasitic activity. Again, providing additional evidence that inhibition of this Plasmodium PI4K was the driver of the host cell potency that we observed. And of course, earlier on, our colleagues at Novartis had in fact shown purely from using a genomic approach that targeting this enzyme is really critical to potentially eliminating malaria. All right, I'm going to switch now to the last part of my presentation to now giving you a story of how we discovered the compound. And the key focus of this is translational medicine. So I've just shown you how we moved from the bench side to the bedside, underpinned by scientific insight. Now I'm going to show you how we moved from the bedside to the bench, underpinned by clinical insight. And that clinical insight, in the context of this program, was what we saw during the human phase 1 clinical trials. This is really in healthy volunteers. And working with our clinician colleagues, What we saw from the clinical studies was this quite inconsistent exposure levels of the drug in different volunteers, which suggested, and not even surprising, because we knew that the solubility was not optimum. And so what we identified as an issue based on what we saw in the clinic was that the key differentiating criteria for a backup program is to really improve the solubility so that we don't have to suffer the consequence of inferior solubility in development. However, what we did in parallel, in order to proceed to really try to understand whether this novel target that we identified is clinically validated, we had to develop a formulation that allowed the phase 1 trial to be conducted and to achieve consistent exposure levels across the multiple volunteers, including what we did with the volunteer-induced blood stage malaria infection study. So we went forward with that formulation and went forward with that formulation into phase 2 human trials. But for the backup program, we needed to come up with something that had the advantage of better solubility. And this is what we did here. And this is a long story, which I'm going to cut very short. So we went back to do several medicinal cell-based optimization to do additional structure-activity relationship studies, which really summarized in a range of publications that we've put out already. But the key data really came from modifications to the core scaffold here. So introducing a second nitrogen from a pyridine to a pyrazine was really a game changer, both in terms of potency, but also in some of the analogs, also solubility. Just to give an example of some of the compounds from the series, not to bore you with the details here, we could see that this change from a pyridine to a pyrazine delivered compounds with very good in vitro potency. You can see very potent compounds, both on the sensitive and resistant strain, but also very impressive in vivo efficacy in the humanized, in this case, in the rodent malaria mouse model study. But while we could see this improvement, in some of the compounds, the cardiotoxicity risk as signaled by the hERG inhibition data, which was variable, we still saw that solubility remained a key parameter to optimize. So we did additional work in other additional analogs. And you can see with these compounds summarized here that we actually managed to really solve the solubility issue without compromising the other desirable properties, including potency. And I would like to single out this compound here, apart from very good solubility, but also very potent in vivo efficacy at quite low doses. The other compound I'd like to single out is this carboxylic acid here. Also reasonably good solubility, but also very high efficacy in the mouse model. Did this good solubility, did this good metabolic stability translate to in vivo efficacy and in vivo pharmacokinetics? And that's exactly what we saw here, that we could see the recapitulation of the good solubility, the good metabolic stability translating to good in vivo efficacy. You can see with the ED90, this compound here, in fact, virtually all of these analogs here delivered exquisite efficacy in the humanized mouse model with less than 1 milligram per kg ED90 value. Very impressive. but also very impressive bioavailability, for example, in this carboxylic acid and this amide derivative. Okay, how are we doing with the life cycle stage profile? And this is just a head-to-head comparison of the compound 048 that proceeded into phase 2 human trials and the backup compounds. Although the focus was on improving solubility, in the process of improving solubility, we also improved on other properties. So without boring you with the details of what's on this busy slide, we have a much more potent compound on the different life cycle stages of the parasite, the liver stage, the blood stage, and in transmission blocking. Much more potent than the front runner compound, even though that wasn't necessarily the initial focus of the program. It was simply solubility. But whenever you are making changes to a structure, it's very important to make sure that you are still on target. But you can have a mechanism switch with very simple structural changes that you make to a compound. And so we have to make sure that when we delivered the compounds in the pipeline, we have to make sure that we are still on target. And everything I described before in terms of how we identified the target, from the genomic approach of doing resistance selection to the chemoproteomics approach, to also using biochemical assays to see whether there's a correlation. All of those things were precisely recapitulated in these analogs as well. Again, confirming that we were still on target with these compounds here. The same thing we did with the other compound that went into phase 2. Without me boring you, we did the same toxicity studies, the same pharmacokinetic studies in rodents, in dog, in monkeys, which was really important in doing human dose predictions, which in this case was far superior to the compound that went into human clinical trials. Because in this case, we had much greater potency, much superior pharmacokinetics, which really fed into having a much lower predicted human dose. This compound, after going through all of these essential, extensive safety studies, was eventually also approved as a preclinical candidate in 2016. So, what are the takeaways? And this is my final slide. What are the takeaways from what I've just described to you? These are the takeaways. So I've shown you right from the beginning of a phenotypic wholesale screening campaign where we didn't know the mode of action, and we didn't even want to know at the beginning of the program. The focus was exclusively on identifying a compound that was efficacious and safe based on our target product profile, which informed our screening cascade and therefore our progression criteria. So that effort in the value chain you see here, from the hit all the way to the optimized lead, that process was really facilitated in this case. And if I told you how many compounds we made, as they say, in drug discovery, you have to kiss many frogs before you meet the prince. To arrive at the compound that went into human clinical trials, we made more than 400 analogs. But if you look at the starting structure and the final structure, there's a very small difference, but that's science. And to identify the backup preclinical candidate, we made about 310 analogs in the program. I've already made reference to the optimization and how that proceeded. But importantly, we delivered 2 preclinical candidates, one of which proceeded to be a clinical candidate and reached phase 2 of human trials. And importantly, this process, this drug discovery program, allowed us to identify a novel malaria target that the parasite has never seen before. And this target, PI4K, is clinically validated because the compound reached human clinical trials and showed efficacy in patients with malaria. So this is not just a contribution to basic science. It is also a contribution to clinical science. This represented the first time in which a chemical proteomics approach was used to identify a malaria drug target, but also the first time that a kinase inhibitor for malaria was tested in people, but also the first time that a lipid kinase inhibitor, in this case PI4K, was tested in people, therefore really providing the most important validation of a drug target. To finish off, this is an effort that involved many teams and many organizations across the globe. And you see this reflected here in the acknowledgements slide that I have shown here. Many thanks to every collaborator here. Many thanks to our funders that gave us the funding. And then, of course, we in Africa love proverbs. We love proverbs. This is a proverb that I am very fond of. It says, if you want to go fast, go alone. If you want to go far, go together. And I'm sorry if I offend Manchester United supporters. I am also a Liverpool Football Club supporter. And in Liverpool, we say, you will never walk alone, because that's what partnerships is about. Finally, we live in one of the most beautiful cities in the world. Cape Town is not just the mother city. Cape Town is a promised land. And that's why I have been here the last 26 years. This is the campus below the slopes of Table Mountain, and I'm in the building very close to the mountain. You can really be envious of me right now. And of course, this is a picture you see from another angle. I am very close to the mountain and very close to the ocean, very therapeutic. Finally, when you come on that flight, this is how it looks like from the top. And if you are interested in soccer, this is the stadium, the Soccer World Cup Stadium here. We play soccer, but we also play rugby. With that, thank you so much for your attention. And Martin, back to you.
Thank you very much. Let me share my slide. We have a few questions from the audience. And after that, we'll get into the panel. So actually, Kelly, it's very interesting. We got a question from South Korea, but from a Kenyan. Alex Munyoki is in South Korea from Seoul. He says, very inspiring talk. With challenges in solubility, did you investigate the effect of co-crystallization or polymorphism to improve solubility?
Great question. Thank you very much. No, actually, we didn't, mostly because what we often would do is when we went to the formulation of the MMV048, which went to the clinic, we could see very consistent exposure levels irrespective of the batch of the material. So when we scaled up, often polymorphism, when you get co-crystals or you basically, when you get these polymorphs of crystals where the crystal packing is different that affects solubility, then you can get very inconsistent results. But we didn't really decide to improve solubility using co-crystal approach, which we have done in other programs where we use co-crystal formation to improve solubility. But in this case, we managed to find a formulation that gave us consistent exposures.
Okay. Very good. The second question is from Bin, who's in Beijing. Professor Kelly Chibale, thank you for sharing this amazing and surprising work. I have a question for how you collaborate with people in traditional medical field. This should be a tough job.
Definitely is a tough job. So first to be clear, so when I used traditional medicine on my slide, it was more in the context of the way people think about medicine is that you focus on a pill and you focus on treatment. But I think that this question is really more about African traditional medicine, just like Chinese traditional medicine, is how do we engage with people like that. So first of all, of course, one approach to drug discovery is, of course, to look at nature as a source of starting points for drug discovery. And I think I am of the firm belief that natural products really occupy an important place in drug discovery. And I think people involved in traditional medicines, I think we have to be respectful of cultures. We must be respectful of what people believe in and what they do. But the challenge for us scientists is really twofold, is how do we reach out to traditional medicine practitioners so that we can provide scientific information that they need to know that when they are treating patients with their products, given that the medicines that we take today, let me give an example. I'm an African, and you heard from Martin's introduction. I was brought up in villages, remote villages and townships, or slums, as they call them in other parts of the world. The diet then, the African, me as the African today, and me as the African 40 years ago, I am very different. Those days, a six-pack came naturally. Today, I have to suffer in the gym to get a six-pack. I don't even have it. I'm trying, but I keep trying. But the point is, we cannot continue practicing traditional medicine today as we did it previously, because the diet has changed, because we have new diseases, new drugs. So we have to reevaluate the safety of traditional medicines. Secondly, and finally, we have to build trust with communities. And that trust comes from being respectful, but also understand that we are partners, that we can bring intrinsic value to traditional medicines. I think they should be used as they are. Most of them are necessary to be used as concoctions. What's important is providing safety information. Those products must be tested clinically. And when we have clinical data, we can speak about safety and efficacy. At the moment, that's in the dark. And one of the problems today is liver-induced damage, drug-induced damage, because people are still taking traditional medicines as they used to. And yet most things have changed, including diet.
Thank you very much. The second question is from Jingchun from Shihuan. Congratulations for your great achievements. Did you use this method for other diseases and drugs?
Thank you. If I understand the question, this method, the approach, yes, we have used it in TB, and we are using it in AMR as part of our Johnson & Johnson Center. In TB, in fact, this is the dominant program, but we also have managed in a number of cases to deconvolute the mode of action, where we understand what the target is. And then we use both a combination of cell-based medicinal chemistry optimization, but also target-based optimization. So we can always check that we are on target, because we've seen in a number of cases, especially in TB, where we've seen many examples where we have a mechanism switch that we just need to make sure that we develop the biology tools to interrogate everything that we're doing. But yes, thank you for your kind words. Yes, we have used this approach in TB as well successfully, and we have made also very good progress in TB. Thank you.
And thank you very much. Those are all the questions from the audience. So let us now go to our panel. And first, I want to introduce Adrian from Rhodes University. Adrian is an associate professor of biochemistry She holds the Department of Science and Technology, NRF South Africa Research Chair in Molecular and Cellular Biology of the Eukaryotic Stress Response, and also a Newton Advanced Fellowship of the Academy of Medicine, Medical Sciences in the UK. She's a very, very prolific scientist, done some great work with her interest being on cellular protein homeostasis in physiological and pathological conditions. She has multiple awards. I won't go through all of them. They're quite a bit. She's been awarded the Rhodes University VC Distinguished Research Medal in 2015, the DST South African Women in Science Award for Distinguished Young Scientists, and she holds a C1 rating with the NRF and is a fellow of the African Academy of Science since 2018, Cell Stress Society International, and is also a member of the Academy of Science of South Africa. She's also in several editorial boards, and most prominent of which is in the Scientific Reports, which is a Nature journal. Our next panelist is Professor Ndikoma Olugbem. She is an assistant professor of cell biology at the Faculty of Pharmacy of Bamako, University of Science, Technique and Technologies of Bamako, USTTB in Mali, in West Africa. She received her PharmD in University of Bamako in 2004 and her PhD from UPenn here in the US in 2014. Her research laboratory is based at the Malaria Research and Training Center in Mali, and her work focuses on the biology of the malaria parasite development and malaria transmission in African patients. Then our ex-challenger is John Woodward. John is a chemical scientist with interdisciplinary experience and a passionate interest in developing molecular strategies such as treatments and tools to tackle the scourge of infectious diseases. He received his PhD from the Department of Chemistry at UCape Cape Town in 2016. Is a junior and is currently a junior fellow at the Holistic Drug Discovery and Development Center, H3D, that we heard about at UCT. His research work is in medicinal chemistry and chemical biology aspects of malaria drug discovery, and he's very, very passionate about it. So without further ado, let's jump to the panelists. And as is the tradition, we give the floor to our ex-challenger.
Yeah,
John?
Thank you so much. Thank you very much, Kelly, for a most inspiring talk that really demonstrated the promise of translational medicine and drug discovery in bringing new medicines from the bench to the bedside. So thank you. And my role as ex-challenger is to represent young and early career scientists. So my questions are going to be a little more general in scope. You showed from your slides, the value of hard scientific skills, things like laboratory techniques, data analysis in deriving important scientific conclusions. But what do you think are the most important soft skills that young scientists should focus on today?
Thank you very much, John. Yeah, soft and not hard skills, because hard skills are obviously for the lab. And I think that perhaps my own story can illustrate this. As you know, John, I am trained as a hardcore synthetic organic chemist, and the audience picked up from Martin's introduction. I was trained in synthetic methodology and photosynthesis. But my career has really evolved, and I think when I started my career in Cape Town 26 years ago, moving from Scripps from San Diego to here was the reality of what confronted me. And so, if I can use the issue of soft skills that you asked me about, I think first of all is keeping an open mind is really, really important, because so that is a skill. Actually, keeping an open mind is a skill, because when I was confronted with the reality of the fact that I couldn't do photosynthesis in the way that I could do it at Scripps or in Cambridge, because of the reality of the environment here, and so I had, as Martin was echoing from the introduction, I had to reinvent myself. So part of it is to have that keep an open mind and, focus on the opportunity that's in front of you, to be able to keep an open mind about opportunities, because at the end of the day, we're all scientists with certain skills. Martin is a materials scientist, and so he must stick to that. I am a synthetic chemist, I must stick to that. But it's also about having the ability to use chemistry in other contexts, in biology. So today, sometimes when I'm speaking, people think I'm a clinician, and when I'm speaking, sometimes people think I'm a politician. No, it's because I am willing to learn, and I'm continually learning. So I think it is realizing that you have a role to play in everything you do, and you need to be open to those opportunities, and to have the personality to get along with people. That is an important skill. If you cannot get along with people, I don't want to work with people who are a pen, for example. Just having a good personality, being friendly is a good example. How do you make friends? You start by being friendly. How can if I go to a conference, I see somebody looking really miserable with a grumpy face, I'm going to turn away from it. So even just being friendly, those are simple things that go a long way in building relationships.
Thank you very much, Kelly. Yes, I must agree with you there. And thanks for sharing those insights. Certainly does make for a nicer work environment as well. And you want to come to work every day when your colleagues are friendly and cheerful and a pleasure to work with. So perhaps just sticking on the topic of people for a moment, mentorship is very important, particularly in the scientific field, not only to provide sort of role models, but also so one can see what is possible. So may I ask who your role models have been, your mentors have been over the years, and not only in a scientific context, but personally as well? And what are some of the values or characteristics of those role models and mentors have really been important to you?
Thank you very much, John. So I'm going to separate the two. On one hand, it's my mentors. On the other, it's the role models. So let me answer the easiest one. And this might disappoint you and disappoint the listeners. My role models, and I'll come to mentors in a second. My role models is anyone who is humble. You are my role model if you are humble. No matter where you are, full stop. Those are my role models. Humility. Mentors, I cannot just underestimate the impact of my mentors. You spoke about role modeling. There's power in role modeling. When people watch you consistently, the things you say, your actions on a consistent basis, what you have achieved, they get inspired. So role modeling is just there. You don't even have to preach it. You don't have to say it. It speaks for itself. And it's really my mentors myself, starting with Stuart Warren. When I moved from Zambia and the audience heard Martin's introduction, I was disadvantaged. Everything was strange when I moved to England. The weather was strange, strange weather. The food was strange, strange food. The people were strange, strange people. The science was strange, strange science. But this man took me more than just being a mentor, but a father figure, to the extent, just to give you 2 examples, how much he cared about not just the science, but me as a person. If I mispronounce a word, for example, he took us to a restaurant in the UK. So one thing about the UK is, it's British manners or British diplomacy. So we go into a restaurant with, some of my English friends, and we go to this restaurant. People are choosing menus. I didn't hear what they were saying. So when it was my turn to choose something to eat, I just said, "Oh, I want," and I pointed. And then he just bended backwards and whispered in my ear. He said, "You say please." And if I mispronounce a word, I used to say, this function group called Esters. I used to say Easters. He would say, Easter is a holiday. Yeah, so he cared about everything, even the way that I pronounce words. So a real mentor accepted me for who I was, and he was prepared to work with me, didn't write me off. all the way to Nick Greaves, who I did my postdoc with at Liverpool, a walking computer. Nick taught me something else that I didn't get from Stuart, very different. And of course, when I went to Casey Nicola at Scripps, really taught me incredible things about passion, drive, creativity, and how you can build a big group and succeed. So those are my mentors and my role models. Is the people, my PhD advisor, my postdoc advisors, Nick Greaves, Stuart Warren, my PhD supervisor, and Casey Nicolau. Very grateful to them. And if they are listening to me right now, thank you, Casey. Thank you, Nick. And of course, the late Stuart.
Thank you, Kelly.
Okay.
If I may ask maybe just one more quick question, just about the balance of hard work, but luck in science and in drug discovery. You spoke about kissing many frogs. You had to make many molecules, test many molecules to find the right one or the best one. What role does luck play? Do you think you create your own luck through hard work, or are you just sometimes in the right place at the right time?
No, I think you create your own luck, if I can use that way. It's actually, it's more, the more that you try, the more, then of course, miracles happen. So when we discovered by the time in 2012 that we were declaring the MMV048 as a candidate, that was within 18 months. The typical industry standard, of course, things can be quicker or slower, is 6 years. So it happened in 6 months. But I think the biggest lesson, John, that I learned is often it's simply looking at the data that you have. We are so driven to, this is especially the chemists, we like formulas, we like to, or if it doesn't fit Lipinski rule, or it does, we like this, we don't like to take responsibility. But I think what I've learned is also just going back to what you've already made and reevaluating what you already made, going back. And of course, all it takes, by the way, is one champion in the team, because there was just someone says, hang on, there's something about this compound that we missed initially, So, but I think it's hard work, and you create your own luck through hard work. It's not being lazy.
Thank you, Kelly. I'll be back in the lab straight after this talk.
Very, 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 very interesting. And Kelly, you have so many insights there that I think our audience will be salivating to really listen to what you're saying. Then we shift to our panelists, Adrian or Ndikoma. Any of you can jump in.
Okay, I can maybe start. Kelly, thank you so much for a wonderful talk and a tour de force from this molecule all the way to the most amazing biological analyses. Perhaps I can ask you about the approach you use to develop the resistance. So developing resistant parasites and then, coming up with the whole genome screens and the single nucleotide polymorphisms. So does that data have value beyond, essentially just identifying the target? Do you have any plans to use that information to try and identify what the mechanisms of resistance will be, how great the resistance might be, that sort of thing?
Thank you, Adrian. Yeah, so one of the things that we actually have done with that particular mutant line and others that we've generated on other series is that actually creates a panel of resistant mutants that we actually then deploy in new drug discovery programs to, when we front load those kind of biology studies, what we then do is that we can first of all establish whether any new chemical entity is cross-resistant, even if it's working by the same mechanism. If there's no cross-resistance, it just suggests that maybe you're getting mutations elsewhere. And so even if you have two compounds that are going after the same target, if they bind differently, then of course that's it's a novel mode of inhibition of an existing target. So to answer your question, we use these mutants that we generate from resistance selection partly as a mechanism to triage new chemical entities, either that we want to use that are not based on existing mechanism. So we said we don't want anything that works by these mechanisms. Okay, how do we check it on the resistant mutants that we generated? And then that also helps us to rank chemical series if we want to prioritize one over the other and focus on those with potentially novel mechanisms.
Great. And I mean, do you see, have you done any analysis at the level of the protein? Do you see whether these mutations change the biology of the protein that would then explain the acquisition of resistance? Or is that too sort of tangential from the drug discovery component?
Yes, Adrian, it is tangential. We haven't done that investigation. Yeah.
Because I imagine if you're focusing on delivering the next candidates and whatnot, that's probably an interesting academic subject, something of interest, but perhaps not the major priority in terms of going forward.
Yes, I think this is, as you know, at the university here, I wear two hats. So I have an academic group where I have PhD students and postdocs, and of course, it's drug discovery. And I think the fundamental difference really between the two, that in academia, the project belongs to the PI, and the PI milks it for all it's worth. It gives them a reputation, whatever.
Yeah.
And of course, you answer interesting questions. You publish amazing papers in Nature, in Angavanta, wherever you publish. But drug discovery, the project belongs to the team, and it's about decision making to stop something that is not working. So, as they say, you kill fast. So you kill cheap, as they say. But it's also that it's hunting for drugs. I actually don't like to use the word drug discovery. No, it's drug hunting. A drug hunter is very different from a drug discoverer. So we do beautiful. John is an example of that. We do beautiful chemical biology in the things that we come out of the drug discovery, where we focus on what is on the critical path to a safe and efficacious medicine, because the patient is waiting. The patient is not waiting for a Nature paper or a Science paper. The patient is waiting for a medicine.
Yes, and I think you definitely communicated that in an excellent manner.
Thank you, Adrian.
Ndikoma?
Okay.
Thank you, Andrea.
Thank you very much for this very inspiring talk, and it was such a pleasure to just see all the path. I mean, you just boosted me now. Tomorrow I'm going back to the lab, and I won't be discouraged, trust me. So my question goes back again to the resistance, because for us facing malaria, every time that we have a drug, what we're concerned about is when the resistance will just start. Chloroquine was introduced in 1945, and 12 years later, we start seeing the first strain that was resistant to the drug. Proguanil was introduced in 1948, and just 1 year later, it didn't work, and we start seeing resistance. Sulfadoxine pyrimethamine, a combination drugs, was introduced in 1967, and the same year we start seeing resistance. So when I see all the work you've been through and all the resources that have been used to find this new molecule, so my question just goes toward the resistance. The first question is, from all the start when you looked, did you think about the structure of the molecule? How different should be the new molecule that you want to put in the pipeline? So from all those molecules that you screened, were they different from the one that we're already using? And how different are they? And even when you went back on the lab and tried to design new molecules, did you keep in mind that you want something that is different in terms of structure? That's the first question. The second question, or maybe you can answer to that one, and then I can ask the second one.
Thank you very much, Ndikoma. Thank you. Yes, so one of the criteria for a series, you make either on the basis of chemistry or on the basis of biology. So if it's biology, it's what I was saying to Adrian earlier. So we front load biology triage, where we want to rule out chemical matter that acts on existing non-mechanisms. So that's a biology triage. But the chemical triage is that we want something that is structurally differentiated from what is available in the clinic. Because again, it's all about recognition. You want the organism not to be too familiar with the chemical. These things recognize chemical structures. And so if they are not very distinct, that the fate of that they will encounter, drug resistance, I mean, for goodness sake, it's a natural consequence of chemotherapy. So it's not if, it's when it emerges and how we can slow it down. But we are more likely to delay the emergence of drug resistance at the biology level if we have a novel mechanisms or novel combination regimens, or a molecule that is structurally differentiated from what is available out there. That is my first answer to your question, Dinkoma.
Okay, thank you. The second question is, since from my background, usually we are really at the interface between the bench and whatever is happening at the bedside. It's probably more like a philosophical questions. How can you help you in this quest to kind of assess the presence of strains that are naturally resistant? I mean, if the resistance is already there, because people are using some of the drug, although they haven't been, the current parasites have not been exposed to these coming molecules. How can you help you in this quest as an African scientist working in those two interfaces? How can we help you? Thank you.
Thank you, Dinkoma. Yes, so this is exactly why the word here is translational medicine. It is really working with clinicians who are in the field. So when we talk about resistance, so we can generate resistance in the lab and resistance strains in the lab. And we can use them partly to triage molecules or chemically or biologically. But also we can use those mutant strains, maybe back to somewhere around what Adrian was asking about, to then proactively look for those mutants in the field and see whether they're beginning to emerge. But ultimately, what matters most, and this is, I'm glad you've asked me this question because I didn't even touch on this, is that we had to make sure that those compounds, one of the requirements for their nomination as clinical candidates is to show activity on circulating resistant parasites in the field. And that requires direct interactions with the clinicians who are doing the work. And actually, before we start any of our drug discovery programs, we want to have insight from clinicians. What do the people want on the ground? If you don't talk to clinicians, you will never know where you need to go and what you need to do something that is relevant to the population. That's exactly why this interaction is so important, that I am still learning. And it's very exciting to be able to learn from clinicians and see how they think about these things. Otherwise, it's in a vacuum if there isn't that bridge. So this translational aspect is a bridge between the lab and the clinic.
Mm-hmm. Thank you very much. I think this was all for me. And thank you.
Thank you very much, Kerry, and thank you, everyone. We still have a few minutes, so I'm going to jump in. And when you started talking about PI, phosphoinositol, the first thing that came to mind was the late Brett Fraser-Reed. He advocated very strongly the use of GPIs, glycophosphatidylinositols, as potential vaccine candidates, vaccines, not treatment, candidates for both TB and malaria. And it draws me back to a conversation I had with him when I was still a student, talk about mentors. And he took me through why he was very adamant that those could be potential candidates for this. And tying the question Adrian and Dinkoma asked on resistance, do you see the GPIs playing a role at all in the process of eradicating malaria and TB?
No question about it, Martin. Thank you, and thank you for reminding us of one of my human role models is the late Bert Fraser-Reed, and I was very privileged to meet him. Actually did visit me here in the building, just below the mountain. So he also appreciated the mountain. And actually, I remember that trip, by the way. He was on his way from Kenya back to the US.
Yep.
So yeah, tremendous organic chemist and a tremendous role model for minorities like us. Without question, a number of these vaccine candidates and monoclonal antibodies, for example, biologics. Some of them are really based on these GPIs, and I think this is an area, by the way, that's becoming quite popular. There were just literally 3 weeks ago, there was a piece written by MMV. This is our colleague Tim Wells and Christina Donini from Medicines for Malaria Venture. This is actually was in the New England Journal of Medicine, talking about monoclonal antibodies use in malaria treatment. But as you know, with vaccines, vaccines have been very challenging in malaria because this parasite is really, this is the most sophisticated animal on this planet, is the malaria parasite. And you can see the biggest advance we've ever made in the vaccine field was this vaccine that was approved last year by the World Health Organization in children less than the age of 5, and it is only 40% effective. But that's a significant achievement scientifically, because this is the first vaccine in more than 100 years that's been developed. So even if it's only 40% effective, if you consider children, 400,000 children dying, 40% of that, we save lives. But absolutely, back to the GPIs, absolute room for that. And I think this field is crying out for the kind of technology that COVID, has shown us as well. I think they are now, I think it's one of these, I don't think it's Moderna. I think it's Moderna. They announced that they're going to be, working on, and forgive me if I misrepresent them, but I think some of these companies are interested in this RNA vaccine applied to malaria.
Very good. Adrian, I see your hands up.
Yes, thanks, Martin. If I could engage Kelly with another question, because obviously we're talking here about getting this phenomenal success first in its kind of from the African continent, getting it to these clinical trials. Now, do you have a clear vision for, assuming it succeeds, where it's going to go from here, and how much of a role African scientists are going to continue to play in terms of getting this compound into large-scale use within communities?
Thank you, Adrian. So first of all, I kept saying, the compound reached phase 2. And that's because we stopped with our partners, stopped the development in phase 2 for 2 simple reasons. First of all, retrospectively, we saw in rats, this is after it'd gone into people, saw in rats, in looking at the data again, saw the embryo toxicity. This is teratogenicity that the compound displayed. Now, there are many medicines that are used that are teratogenic, but the reason for dropping is twofold. One of the important target product profiles for malaria is to be able to treat pregnant women. 95% of the cases are in Africa. 80% of those children and affect pregnant mothers. So we don't want to neglect this important population. And so for that reason, we felt that we need to understand, and by the way, it's mitigating risk because it wasn't seen in people, it was seen in rodents, which is important. You've got to be cautious. But the second reason for being cautious is that this is a novel target. This is a kinase target in oncology, in cancer. In fact, if you mention a kinase inhibitor in another therapeutic area, people say, toxicity, it's a liability because it's a kinase. But no, kinases, as we've seen here, there are also opportunities. And people forget that in oncology, in cancer, we go for the maximum dose a patient can tolerate. But in malaria, this is an acute infection. And of course, it's a low dose. So it's all about safety margins. But because it's a novel mechanism, and there are multiple programs across industry and even ourselves, we have multiple programs in PI4K. And to Dinkoma's question, we've focused on molecules that are structurally differentiated from those. We know it's a great target, but because it's so new, people are cautious. We want to understand whether this toxicity is related to the target or other kinases. And I think we've identified 2 or 3 that we think we might want to look at very closely. So we've front-loaded those particular kinases because they're the only ones that were inhibited significantly in the entire 500 or so human kinome that we looked at. So for that reason, because it's novel, people want to be cautious and we want to understand. And it's good when we understand the basis for the toxicity, because then we can address it because we understand it.
Yeah, great.
Very, very fantastic, very nice engagement. And you know, I want to say, Kelly, you know, I hope this is a big shot to every young scientist in Africa and even the established one. Let us keep doing good work. There's so much potential coming out of Africa. And actually, you know, fired me up to start pushing for materials. We don't need to use this excuse that making electronics is expensive. If you can make a drug out of the continent, you can make computers out of that continent. And thank you very much. For the interest of time, I will proceed to the next stage. And Kelly from ICANX, Alice, myself, Fulan, and Paul Weiss, and Chris Nyhouse, and Miso Kim, all the moderators, all the organizers, we want to thank you very much for coming in. And if we were in the actual in-person, Alice and I would walk the stage and present you this beautiful certificate. But now you're going to have to accept a virtual one, and please print it and put it in your collection. You grace our 112th volume. And again, this is something that we are very happy and very, very proud of you, of your work, and all of you for being here.
Thank you, Martin, and thank you to Alice.
Since we are hitting quite a bit of African audience, that we do have Young Investigator Awards coming up. Please, please, please encourage everyone to apply. We want to see a huge participation from Africa, from all disciplines. It's not discipline specific. Kindly encourage everyone to apply, and we want to encourage the next Kelly Chibale in the next generation. So thank you very much. And next week, we are going to a new version of the talk in ICANX. We do have a space that we will be hosting in-person talks. And so our first in-person speaker is going to be Zhongling Wang, but it's going to be broadcasted virtually also. So please tune in the same time and engage with our first in-person speaker, who will be beamed from Beijing. I want to thank everyone for coming, and I want to thank ICANX and all our sponsors for their support. Thank you very much.